Okay, thanks. Big thanks to Ian and to the CDB department for allowing us to participate in these great summer Zoom sessions. We've been thinking about this for a while, and this was a great opportunity. So we're going to split this into 3 parts, hopefully about 15 minutes each. I'm going to start with an introduction, an overview of the scissor, basically who, what, and where. We're going to talk about people, equipment, location, and our general offerings. Then we'll turn it over to Nick McNemee, PhD. He's a senior research specialist within the scissor, and he's going to tell us about all things light sheet and 3 light sheet microscopes that we are able to offer now. And then we'll switch a little bit over to electron microscopy, and Evan Kristofiak, PhD, also senior research specialist, will tell us about an overview of what our scissor EM offers and new techniques that are coming along there. And it's kind of tied together because it's some pretty cool new EM things, looking at being able to use EM for 3D. So we're tying that together with what we already do in light microscopy. Okay, I'm going to share my screen and hope that it works. Go back, sorry. Okay. So the cell imaging shared resource at Vanderbilt, we like to think of ourselves as the Vanderbilt microscopy resource. We have a wide range of light and electron microscopy equipment to offer. And we are supported and funded by a variety of different groups. We have salary support from 5 different NIH centers. We are administered and are a part of School of Medicine and BBS, and we are a BU core. And then, of course, we have financial and space location support from CDB, and then we have rooms from other departments too, notably MPB and Light Hall. We have 11 people, 6 of those are PhD level staff or faculty, and we offer a variety of things. Mostly we acquire new equipment and maintain those microscopes so that they are always available for anyone with a BU or BUMC appointment. We routinely consult with people. We talk through the project and help them choose the most appropriate microscope, and then help them talk through their post-acquisition data analysis as well. Help them think through the best application both for acquisition and analysis. And all of that together, we try to keep our investigators at the leading edge of cell biology and of microscopy technology. So these are the things we're going to cover quickly today. A little bit about analysis and new people that have joined the scissor within the last couple of years. We'll give you an update on the Nikon Center of Excellence that was opened in 2016 with great support from the School of Medicine and the CDB department. And then Nick McNemee will talk about light sheet microscopy here at Vanderbilt, and Evan will talk about new scissor EM services. So what do we have? This is just a summary of the different equipment that we offer across campus. Still, our bread and butter microscope is a point scanning confocal. So we have 7 confocals, 6-point scanning, and 1 Nikon spinning disc, a variety of super resolution structured illumination microscopy and stochastic optical reconstruction microscopy for super resolution that lets us push the resolution beyond the resolution limit of light. Wide field microscopes, specialty microscopes such as TURF for looking at things within 100 nanometers of the cell surface, 1 transmission electron microscope, 2 scanning electron microscopes. Full service EM prep, Evan will touch on that, numerous workstations, and 3 light sheet microscopes. And this allows us to cover a broad spectrum of samples and hopefully most any sample that you work with here. So traditional light microscopy that we've been doing for a long time can image within the hundreds of microns down to not about 150, 120 nanometers. Super resolution came along 10 or 15 years ago and can cover about the same, maybe up to 100 microns or tens of microns, but down to about 10 nanometers with STORM. Electron microscopy goes even further. It is going to be in the 1 to 10 microns, but can go down close to single nanometers. And then with our recent addition of light sheet microscopy, we're going on the opposite end. We're imaging larger things. So whole organs, usually cleared, mouse embryos, Drosophila embryos, and organoids. So into the millimeter range and even 4 or 5 more millimeters in X, Y, and Z. And doing this in a more reasonable time so that you're not having to do that scanning overnight. Here's a list of our personnel. I just want to point out, so this is 11 people, only about half of them are full-time. Others work in other departments or labs as well. The ones with the red star are our full-time people. And then we have 3 new people just in the last 2 years that I want to make sure you know their names and faces. So that's Evan Kristofiak in EM. He's been here since 2018. He came from NIH as a postdoc to work in our EM core. Nick McNemee joined just about a year ago. He had done his postdoc here with Owen McGinnis in MPB, and he is going to be our go-to person for light sheet microscopy. And then Carrie Seidel just joined us this past December. She came from Cincinnati Children's Hospital, where she had worked at the bench in a research lab, and then had also worked in their Nikon Center of Excellence as well. So she was a great addition because she had a lot of Nikon experience as well. Okay, where are we? You're probably familiar with some, but maybe not every room that we have. We are excited that we have finally finished our 7th floor light hall renovation. It was completed about less than a month before we went on hiatus, which was fortunate for us. So we kind of have 3 major suites: the 7th floor light hall suite, the EM prep lab, which is on 3rd floor MCN, and then the Nikon Center of Excellence, which is just below that on 2nd floor MCN. And then we have other single microscope rooms in a variety of locations around campus. The images on the right at the bottom are just showing you the layout of our new 7th floor. It was formerly 742 Light Hall. The main door is in the same location, but now we're 704, and it's a larger space. It goes all the way from that door to the elevators at the north end. If you can see my pointer or my cursor, this is the elevators on the north end of Light Hall. You'll come in the same main door here. There are 9 individual microscope rooms, analysis workstations here in the middle, a prep area, a staff area, and an office. Okay, so all these people and equipment and locations allow us to offer you microscope hours. So these are total microscope hours, not including EM sample numbers, just by calendar year for the last 5 years. And you can see that we were around 10,000 hours per year, and now we're up to 12 or 13,000 in the last 3 years. I just did a calculation for the last calendar year, and CDB accounted for 17% of those almost 13,000 hours. So that's fantastic, and we appreciate your support. And you'll notice a major jump between 2016 and 2017, and there were 3 things that happened then. Sam Wells, who was our previous director, retired in 2017, but he had just been awarded 2 S10 grants. One for the Nikon Turf that was installed in early 2016, one for the Zeiss LSM 880 Airy Scan, which was also installed in 2016, and then of course the opening of the Nikon Center of Excellence in October of 2016, with significant support from the School of Medicine and Ian and the CDB department. And that added 3 microscopes, the Nikon Spinning Disk, the Nikon SIM, the Nikon Storm, 2 analysis workstations, and a tissue culture area. What else comes from our people and equipment? Publications. So CISR routinely has over 100, over 120 probably, publications per year that are associated with or include CISR acquired data. These are just a few examples of journals just within the last year of CDB PIs that were published with CISR data. So we appreciate that. And just a quick plea, please acknowledge the CISR in your acknowledgments, and if any of the work comes from an S10, please acknowledge that grant as well. That is for our reporting purposes and helps us, puts us in a better position for getting additional S10s. So now I want to walk through a few slides of how we are keeping the CISR state of the art. First of all, we are, the COE has been fantastic and I think very popular. So we want to maintain its level of popularity, and so we are trying to keep it up to date. One thing that was done about 2 or 3 years ago with Brian Millis and then Nick McNimmy when he was a postdoc was one of the first users of this was to realize the Nikon Storm is fantastic for super resolution, but it's not going to be used all day, every day for Storm. That's a pretty niche application. But it's a fantastic wide field system with a really fast camera. So they realized that we could do really fast wide field imaging, and in particular, Nick, when he was in the McGinnis lab, was doing some intravital imaging of capillary blood flow in the mouse leg. So the movie on the left just shows you some fluorescent beads flowing through these vessels. And then he was able to do some sophisticated tracking in Nikon Elements to look at these capillary tracts and to do a lot of different measurements and learn about insulin delivery. We also have 2 or hopefully 3 upgrades coming very soon. Ethan Lee has put in for an NIGMS equipment supplement to purchase another spinning disc confocal. It would be for his lab, but it would also be available to scissor and COE users. We're still waiting to hear about that, but that would be funds from NIGMS with some matching funds from CDB and Basic Sciences. And then we have 2 other pieces of equipment that we hope will be installed in the next days or weeks. They have both arrived, and we are scheduling with Nikon right now. One is a new light source for the Nikon Storm, and this will do a couple of things. It is the Aura light source. Most importantly, it will add a size 7 excitation, which has been needed by a lot of people. And also it allows separate control of each excitation laser, or excitation output. As I said, that has arrived. We hope it will be installed within a couple of weeks. And then third, we are also upgrading the camera on the Nikon spinning disc. We have acquired the new Photometrics Prime 95B camera through generous contributions of 7 different PIs. We're calling this a new model. We're calling it the GoFundMe model, where different PIs pitch in a little bit for the greater good of those PIs and all of the scissor. And we appreciate their contribution. And so that will offer speed and sensitivity in a larger field of view, and soon you too can be imaging beautiful movies just like Matt Tiska tweets all the time because he already has this Prime 95B camera on his spinning disc. Okay, and not just the COE, all of scissor, we are always looking for new equipment and new technology and listening to our users and PIs as to what they need next. So we are always looking for equipment grant opportunities. We are fortunate in the last year and a half or 2 years to have been awarded 3. 2 of the Basic Sciences core development grants, both to upgrade some of our EM services. So first, late 2018, early 2019, we were awarded money to purchase a new Leica ultramicrotome for EM, just a newer instrument for sectioning. Then a year later, we were awarded $48,000 to purchase a new AMT camera for the TEM, and Evan will talk about that, and that has just been installed in the last month or so. And then just less than a year ago, we were awarded an NIH S10 grant to purchase the Zeiss Slide Sheet Z1, and Nick will talk about that as well. We have 2 more NIH S10 grants that are outstanding. Last year, we put in for a Zeiss FibSim, that's Focused Ion Beam SEM. Matt Tiska was the PI on that grant, and we are still awaiting word. We're still hopeful. The funding is coming late this year. And then just this May, we put in another S10 for a Nikon A1R HD25 confocal. And you can also see here that cell and developmental biology PIs have been very instrumental and supportive when we put these grants in of being major and minor users. And then also at the same time in May, we had an opportunity to put in for a VA sheet grant. That's their shared equipment grant. Roy Zent at the VA is the PI, and Jim Goldenring was very instrumental in helping us and notifying us about this and helping us put this together as well. So those 2 we would find out about within the next year. Over the last 2 to 3 years, we've also kind of put an emphasis on helping users do more post-acquisition analysis. And so we have several analysis workstations that are available. We have a full license of Imaris, which is great for tracking and volume rendering, any 3D and 4D rendering. We have just acquired a Revis Vision 4D along with the Zeiss Z1 light sheet. So we're just starting to learn about that, but it's great for rendering the light sheet data. We have 2 copies of Nikon Elements that are very popular. And then of course we support PG and MATLAB as well. Over the last year and a half, we've also started doing some scissor workshops just as education for scissor users. We have 4 that we did in the last year before the hiatus. And they are all recorded and available on our scissor website. The Basics of Scientific Digital Imaging by Jay Jerome. Evan did 2 workshops on Fijian Image J, and then I did 1 on co-localization. And we're hoping to do more of these. And then we routinely have vendor workshops as well for analysis or for microscopy. Notably, because of our Nikon Center of Excellence, the Nikon reps, before we were working remotely, they would come on campus every other Friday for open office hours as well. And we can talk about working with them to do that some more. Okay, also during our quarantine and remote work time, we know it's hard for us to do our one-on-one trainings that we've been doing. So this is our plan, and we hope to roll this out very soon, hopefully within the matter of a couple of weeks, some online training videos. Notably, this is something we had been talking about and beginning to work on for years, and now is the time that it's really going to be beneficial. So the plan is that we will have a video that is very similar to what we would give to users one-on-one, and they will watch that ahead of time. Then we'll have a remote training session where I or another staff will sit at the scope and the user will zoom in and watch how we show sample placement and things like that. And then on a third session, we would reverse that. So the new user will sit at the microscope, show us what they've learned, and we will advise from another room. Ideally, we would do that. I could be here in my office and could just pop in for a couple of minutes to help a new user. And the first ones, of course, that we will roll out for this will be the Zeiss 710 and 880 and the Nikon spinning disc because they are the most popular. Okay, we just announced on Friday, we have a brand new website, so check it out. There's the address at the top, my.vanderbilt.edu/scissor. Hopefully it will look familiar to our old website, at least a lot of the same content is there. So I just want to show you a couple things you could see. So here under microscopes, you could click on that and you would see our different categories of microscopes. Then you could choose confocal and see a list of the confocals. And then I could choose LSM 880. I would see a picture, but there would be a lot more content there. Filters, objectives, lasers, whether or not it has live imaging, example images. So you can kind of get an idea of which ones you might want to use. But like I said before, we are always available to have that conversation with you. Also, please note we're on Twitter, so check us out there. We've been there for a year or so. And send us your papers and beautiful images, and we're glad to put them on the website and tweet them to the world. Also on the website, we have a nice little chart here that can also kind of help you walk through which microscope might be good for what you need. This is an example for the confocals, so you can see how many lasers are available, what the magnifications are that are available, whether or not it has live cell imaging, so on and so forth. Okay, so that's just a summary of the equipment that we have, the people that we have, new things we've done in the last couple of years, and things that we're looking forward toward in the future. We are excited about the S10 grant we were just awarded and the new ones that are coming. Nick will talk about light sheet microscopy next, then Evan will talk about EM. And we just want to say we appreciate, from this department especially and others around campus, the continued investment. We are always looking for continued growth, and we believe that equals continued success, both for our core and for all the users of the core. Okay, I'm going to turn it over to Nick McNimmy now, who is going to tell us about light sheet microscopy at Vanderbilt and within the scissor.
Hey everyone, thank you again for giving me this opportunity to talk to you about our exciting new light sheets that we have on campus now. I'm not going to get too much into the technical details of them, but I just want to give you a brief summary of the systems that we have, what they're capable of, and how we're excited to bring them to you. So the first thing I want to do is kind of what makes a light sheet different, kind of just a brief summary of that. And when we think about light sheets, I first want to think about how we do our traditional wide field or our confocal scopes and how the light comes into the system. Typically what we're doing is we're shining the light either from above or below through the entire system, and then we're bringing it to a fine slip, and then we have a focal plane right in the middle there. And then we're seeing our image. However, this leads to a large section of photo damage within the sample. So the idea of the light sheets is, what if we could actually shape that light into the focal plane so that we were just bringing the light into where we wanted to image? And then we could image the sample from above or a different angle, and that would greatly reduce the amount of photo damage caused by the light. And that allows us to really be able to look at live cell imaging and prevent photo bleaching in a much better manner. So what are the systems that we have on board here at Scissor? The first one I want to talk about, and Jenny highlighted, was the Zeiss Z1 light sheet. The Zeiss Z1 light sheet basically brings in, so from either side, you can shape the light sheet into this kind of sample adapter chamber. And then what you do is you shine the light through the sample, and then we can rotate the sample around through the top and then use the objective on the back to image the sample. This is a really great system, and it's really ideal for large samples. So it's capable of imaging cleared volumes, organs, and whole embryoids and organoids. It greatly reduces photo bleaching, as most light sheets do. This system excels. It has a tremendously large field of view, which allows us to capture kind of these larger specimens in whole, and it's actually adaptable to live cell imaging. So just a couple quick specs on the Z1 light sheet. It has a resolution up to about 250 nanometers, and that's the ideal resolution of the system. What we actually will get will vary slightly. And it has a Z resolution of about 650 nanometers. It's capable of going to about 100 frames per second, and its sheet thickness will vary depending on the sample and the light diffraction. And we have a tremendously large breadth of field of view where we can go down to 123 microns to 3.5 millimeters per field of view. And then on top of that, we actually can stitch images within this system together. So just a little bit of example data that we've been able to capture during our training and demoing of the system. And what we can see at the top here is a Drosophila embryo that has been stained with E-cadherin and RFP. And what we're able to do is, this is a max intensity projection, and we're actually able to rotate the sample around within the chamber to get really great views. Another instance is this kind of whole mouse embryo that we were able to image through the system here. And I just want to highlight that because that is a 4.87 millimeter long sample. That is a tremendously large sample for our field of view. And then I also want to bring over your attention over here to this cleared human pancreas sample in which we have GFP expressed within the islets. And we have a tremendous amount of view to not only see the big macro picture, but come down in and actually kind of get a great resolution within that kind of organ. So this data kind of Jenny has talked about is the data analysis that we bring for you for these light sheets. The first one I want to highlight really is Arevis. Arevis is kind of baked in with the Zeiss software, and it is tremendously powerful for imaging in 3 dimensions. Similarly, we have Ameris, which is also a tremendous resource that many of you use constantly, and we have Elements available to look at some of these data too, as well as Fiji and MATLAB scripts that are there for you. So with that system, we're really excited to have that on, and it was a tremendous addition to Scissor. The next 2 light sheets are actually brought to us through the Academic Strategic Plan and the TIPS program. And there's multiple TIPS programs, but this particular TIPS program that was awarded was a combination of Scissor, Cell and CDB, Physics and Astronomy, and Biophotonics. And these groups all came together to form BioMid. And BioMid is under the leadership of Anita, Shane, and Matt. And it also has Brian Millis and John Koza and myself in it. And what they've been able to do is build the next two light sheets that I want to talk to you about, the Lattice and the SOPI. So the Lattice light sheet is a great system in which we are allowed to, we image and shape the beam, a vessel beam, through our first objective, project it down onto the sample, and then we have an imaging objective at a 90-degree angle to it. And we actually place our sample on a 5mm cover slip right in the middle. These are both dipping lenses. And what that allows us to really do is, through the shaping of the vessel beam and then the dithering of that beam, we're able to get resolutions of about 230 nanometers in X and Y and 370 nanometers in Z. It's an incredibly fast system, greater than 100 frames per second. Its imaging depth is between 20 and 100 microns. I want to talk about its field of view. It's between 10 and 30 microns. And what we can really image on a cover slip, on a 5mm cover slip, is effectively about 3 millimeters. So this system is primed for cellular resolution and this department, CDB. So this is some example data that we were able to capture on the system about a year ago or so. And this is the M-Cherry S-bin and the CL4 cells. And what we're really looking at is how the actin moves. But I really want to highlight how well this system, the resolution and in the time frame in which we were able to image on this system. It's going to be a tremendous system for subcellular resolution within scissor and CDB as a whole. So the next system I really want to talk about is the SOPI. And the SOPI is the Scandal-Beak Planed Illumination Microscope. And what the SOPI does is it shapes the sheet of light and collects the excitation through the same objective. And the way that it does this is actually we bring in the sheet of light through the side and project it down at a 45-degree angle. And then as you can see here, what we do is we rock that sheet back and forth across the sample. And that allows us to image the sample with really fine detail. Now, the SOPI is really ideal for small model organisms, intravital imaging, and ex vivo tissue. Again, like all the other light sheets, the Lattice and the Z1, it greatly reduces phototoxicity. The thing about the SOPI is it's very, very fast, and it has a very large field of view. So just some quick specs on the SOPI. It has a maximum resolution of about 1 micron and about 3 microns in Z. It can go greater than 100 frames per second. We can image ideally up to about 300 microns down. Our sheet thickness is variable. We can change that, which allows us to change the resolution slightly. And the field of view is about 300 by 300. And that system is wonderful for looking at whole organs and tissue explants. So what we're looking at here is some lung explants given to us from Aaron Pelosa's lab. And what I really want to highlight to you is the fact that this was taken over 92 hours of imaging. So what we're looking at is lung, some lung matrix, and then some type 2 cells. And what they were interested in looking at was, how does inflammation change the movement or lack of movement within those type 2 cells? So it was really, really impressive, and we were really happy with the system to see that we were able to image consistently for 92 hours with minimal bleaching. And that really highlights where the SOPI excels. So the next part I want to talk to you about is these systems generate a tremendous amount of data, and that data needs to be analyzed. So what has Biomed really done is they've generated a 100 terabyte storage system to actually be able to store and have that data located in-house. And then they've connected it with a 40 gig fiber optic network. And that network is great because if we can't move our data around, it doesn't really help. And then they have 2 fully loaded analysis systems. Then those analysis systems are completely available to everyone via remote processing. And they have a seamless integration, so you don't really feel like you're not sitting at the system, which is tremendously important if we're going to talk about image processing remotely. So Biomed also allows us to have Anaconda. It has a copy of Elements, Amira. It also has Arevis, Imaris, Fiji, and MATLAB. So I just want to briefly talk about these systems kind of now that we've seen them, just have a quick chart and the comparisons between the two of them. I really want to highlight the ideal sample that's available to these systems. So for the Z1, it's really a large organoid or organ, a tissue slice, or things like that, live or fixed. The SOPI is really great if we're thinking about long duration imaging in live tissue or live tissue explants. And then the lattice light sheet is really set up for, ideally for live cellular imaging. And now these systems, I'm happy to report, I just finished my training on the lattice last week. These systems are all available to the community and CDB now. The Z1 is available as an image for hire as we kind of learn that system and get used to it. And then the SOPI and the lattice are available currently starting this week. We're opening them up. And the lattice and the SOPI will be coming over to Scissors Light Hall space shortly as soon as we get those done. And what's exciting about that is Biomed is bringing on 2 extra microscopes, the SOPI version 2, which is going to target hopefully subcellular resolution, and then the lattice version 2, which is going to have 2 cameras and allow us to really image two different channels at the same time. So with that, I want to bring it over to Evan so he can talk to you about the wonderful things going on in EM.
Thank you. Let's see, get my screen shared for you all. All right, so Scissor EM deals with all things electron microscopy. We have both scanning electron microscopy and transmission electron microscopy. And we are a full service facility. This means that we will, if you want, we'll do everything for you. We can take your sample that's fixed and get it all the way to imaging on the SEM or all the way down to imaging on the TEM. If you'd like, we can also train you, not right now due to COVID, but we are working on some remote training opportunities as Jenny discussed earlier. Also of note, over the last year, we've kind of optimized a lot of our protocols. So if you haven't used us in a while, I think you'll find that both in scanning and transmission, you'll get a nicer result than you have in the past. A lot of this is developing tissue or cell specific optimizations. So for scanning EM, this is a technique that's primarily for looking at the surface of samples. And sort of its highlights is it has a tremendous range of magnification available. It's based on a raster pattern, so how large we raster the electron probe determines the magnification. And I put some examples here so you can kind of see the range that's available. On the left we have a tip of a villus, and on the right we have a single bacteria on the surface of a cell. Just to give you kind of an idea of the range of what's available. And this can all be imaged on the same sample. It's just changing the raster pattern, like I said. And the other big advantage of the SEM is it's relatively easy for new users to use. So this is something you can learn in a few days. The sample preparation is something you can learn and do in a day, which is different than the TEM. The TEM is more for really high resolution things and looking within cells. So it's typically images of projections through thin sections. When I say thin sections, 70 to 100 nanometer sections of tissue. And what we'll be able to see with this is the kind of like cellular organelle things, macromolecular complexes, for example. Here we have a tough cell. You can see the actin protrusions and these little rootlets that are coming into the cell. And what I wanted to emphasize is we just got this new camera. This is a modern CMOS TEM camera, which is replacing a very elderly at this point CCD camera. And what this camera really gets us is a couple things. The major one being it's extremely fast, and this means that we can acquire images without any drift. The end result of that is we get extremely high resolution images from the same kind of samples we had before. For example, here we have microvilli in cross-section, and these dots here, as you might be able to see, these are individual actin filaments that we're now able to resolve. The other big bonus to this is it interfaces with a modern Windows 10 workstation, And this means we can offer some new services. This includes montage and tiling, which I'll show you in a minute, and also automated tilt series acquisition for 3D tomography. The other big bonus, especially with COVID, is that we now can offer remote access to this TEM, which means that training should be a little bit more simplified, and in general, it should be a little bit easier to use. So let's go over some of these features. First one, automated montage tiling. Oh, the other thing I should mention, this camera is very, very new. It was installed in the last few weeks, and so you're going to see some calibration data here. So montaging and tiling is the first one, and this is a way of essentially taking a lot of frames and stitching them together to get a very large field of view. The TEM traditionally does not have a very good field of view due to its camera and a few other limitations that it has, but we can use this tiling method to get a huge view. If you look here, this is actually the same image on the left and right. I just blew it up in the square area. So here we have a 1 micron scale bar, and I just blew this up and to show you that we still have resolution. In fact, our pixels are 5.5 angstroms. So if you go from sort of micron scale bar with 5.5 angstrom pixel size, you get an idea of what is possible using this technique. The other thing we're able to offer is tomography. We were doing tomography before, but now it's automated and much easier to use. And this is an example from the Wasserman lab. So they're interested in these caveoli here. And in 2D projection, there are so many caveoli in these endothelial that they kind of overlay each other, and it's very difficult to analyze them and figure out what's going on. So tomography is a way of getting 3D data out of these sections by taking images at many different tilts. And the end result is here. So the image slowly changes, and it's small changes because these are caveoli, but you can sort of follow them now and see individual caveolae and how they're interacting with other ones. I'll let it just repeat a couple times so you can kind of get an idea of what's going on. So now we have this data and we might want to analyze it. So the next thing we've been working on is starting deep learning segmentation. This is still very much under development. It's not ready for prime time yet, but we're getting there. And so the idea here is, rather than having a graduate student or postdoc sit and essentially segment out all these membranes to analyze the data, we can actually teach the computer how to do it. And this is the result we have so far. It's pretty good. We got all the caveoli segmented. There's just a few discontinuities that we're working on still. And I expect in the future we should have this ironed out real well and ready for users. The next thing I would like to talk about is free substitution. So this is something that we've gotten working pretty well over the last year. So in conventional TEM preparation, these are sort of the major steps your sample kind of endures. So fixation, post-fixation, and then we have to actually remove all the water from the sample to put it in the TEM. This is dehydration, infiltration with a resin, and then polymerizing the resin at a high temperature. So if you think about your actual sample, here I just have an example of a protein I used to work on, Cloudin. And just to show you how hydrated it actually is, these are all water molecules on the top and the bottom, and this is the membrane. So each of these steps, we're actually removing all that water, which causes the protein to misfold. The dehydration causes us to lose some of the lipids, and then adding a resin also causes a lot of problems with the lipid membrane. And then further heating up the protein to 60 degrees also causes some denaturing problems. And this is one of the problems that we have with immunolabeling too. When you denature your protein, the antibody can no longer recognize it. So what we've done is implemented something called free substitution. So this is where we do all these post-fixation, dehydration, infiltration, and embedding. We do all these cold. So we fix the sample, we freeze it in vitreous ice, and then these steps are all carried out at -90 degrees. And the idea is that the lipid and the protein is sort of locked in place due to the temperature. And so we minimize the artifacts that are generated during this process. We also embed at minus 30 degrees C using UV instead of the heat. And this also helps the proteins maintain their normal conformations. I have an example to show for this. This is from a few years ago, but these are our stereocilia. On the left, we have stereocilia that were just prepared in a conventional way at a different core facility. And you can see that the membranes are a little wavy and the actin is somewhat hard to resolve in some parts of it. And this is because the membranes got deformed during the process. And on the right, this is free substitution. And I think the major thing that might jump out is that these round membranes are now very visible and the actin cores are quite visible as well. So this is something we can offer now. It's a little bit different process, but the end result is fairly similar, except we get better results in general. And this is something we can consult with if you're interested in. And now I'd like to kind of highlight some studies in the last couple years and some places we're going with them. So this is a paper from the Golden Ring Lab and the Tisca Lab, and they were interested in imaging inclusions in enterocytes. So these are actually microvilli inclusions within the cells. And they imaged them in two ways that I think make a lot of sense. They used the LSM 880 ARI scan, which allowed them to see the inclusion and some stuff that came in with the endocytosis. And here we have just a conventional TEM showing microvilli within the cell. Now both these are very useful, but they don't give us a full 3D image of what's actually happening. Like how are these microvilli packed inside the cell? Are they just randomly packed, or they look more like the surface of an enterocyte, for example? And this is where I'm going to bring in the SEM, which is normally thought of as a tool to look on the outside of cells, but it can actually be used on the inside too. So here, if we look at what these look like in the SEM, what we've done here is we've taken a piece of intestine, we fractured it so we can see inside the cells. So we have inclusions that you may be able to see here. I'll increase the MEG so you can see them a little bit easier. We can even take it a step further. And here we can see the microvilli, we can see their lengths, how they're packed together, get a little bit more information on what is actually going on within these inclusions. And in fact, they actually, microvilli look pretty normal, despite the fact they're inside a cell rather than on the surface. And now I want to highlight another paper. This is from the Weaver Lab. And they took two approaches that I think are kind of clever and things that I think we can offer everyone else in CDB. So Bong Hwan did most of the work here. And what he had is a very cool system where he has endosomes that have a pH-sensitive GFP on them, so that when the exosomes are excreted out of the cell, that we can actually see them. And he applied CLEM to this, so he was able to correlate light microscopy, which is here, to scanning EM up here with very high precision. And this is something that he largely did on his own. We taught him how to use the SEM, but he was able to do this. I guess my point here is that you can also learn how to do this. And CISR in general is in a very good position to do correlative light and electron microscopy. We have many microscopes that are capable of imaging this kind of GFP imaging, and we can correlate this well with the SEM. And then the last thing I want to highlight is this immunonegative stain. So this is traditionally a very difficult technique to immunolabel gold onto a sample. And here we've combined immunolabeling with negative staining, and we came up with a protocol where we can actually do an anti-GFP in this case onto the exosomes. And it's just another way of getting an immunolabel to actually work in EM. And with that, I'd just like to thank everyone on behalf of Jenny and Nick and myself. And I think we'll take some questions.
Thanks so much, Nick and Evan. yeah, we are available for questions. We have several minutes left.
That's great, guys. Thank you.
Thank you.
Questions?
Hi, Evan. This is Alyssa. I guess I have a question about the freeze substitution and immunolabeling of the samples.
Oh,
Could you
sure. Yeah.
comment a little bit, maybe elaborate just a little bit more about, the possibilities of that? Because I know that that's something that had sort of held us back in the past.
Yes. So immunolabeling sections is traditionally pretty difficult due to the proteins essentially denaturing and the antigen site basically not being accessible to the antibody. The freeze substitution process can help with this. I've gotten it to work many times in the past where the protein keeps a lot of its structure enough to where the antibody will work. It's not 100% guaranteed that it will, but it's much better than the chances we've had in the past.
So I actually just had a quick question about the MATLAB that you guys have. Is that it's just that you have MATLAB on those workstations, or are there scripts written in MATLAB already available for doing image analysis?
So the MATLAB is just available, yeah, like you're saying, through the traditional university license. And then what that allows us to do is Imaris and Elements through an export function. You can write your own analysis protocols. So one of the things Jenny had shown from the image tracking was in MATLAB, with the help of some engineering help, I was able to actually write a script that helped me kind of better track and get some of the more fine-tooth data. So for the analysis software, it's kind of really helpful to be able to have that flexibility to bring data over into MATLAB, process the data, and get things that you wouldn't be able to get out of an Elements, Imaris, or Revis.
Okay, great.
Can I jump in with one quick? So with the deep learning, I guess for Evan, is this, how much training does this require from our individual samples, or should this just be ready to go when we plug in a worm, for example?
It will definitely need some training. The example that I showed has 10 slices of a tomogram trained, and then the rest was done on its own.
Okay.
It'd probably be very sample specific how much training it requires. However, once you have it trained, as long as we don't change the magnification or anything like that, it should just work.
Hi, I had a similar question about the 3D model. Is that one, so is that mini slices, or is that just from one slice? I missed that part.
So tomography will generate many slices. It's the whole section. So TEM requires sectioning. What we're doing there is kind of subdividing the section into many slices.
I guess that's what I meant, is that one, so it's just from one section. Can you
One
make
section, yeah.
that section standard thickness? Like
We can do them a little bit thicker. So I think that one was about 100 nanometers. We can probably push it to 120. After that, we'd have to go to like serial sectioning or something like that to get a bigger volume.
All right, thanks.
Jenny, this is Mark. I just wanted to ask about consultation related to the use of the, new microscopies, all the fluorescent microscopies. Are you available then to help guide users as to the particular application that best suits their needs?
Yes, we're always available to do that. I mean, even if you've never done any microscopy before, just email one of us and we will talk through your experiment and help you choose. If you're specifically asking about light sheet, then Nick's going to be the major point person for that to help you choose which light sheet might be best and make sure your sample is appropriate. And we can do that by Zoom or phone calls right now, if that's your question as well.
Anybody else? Any other questions? Okay. Well, thank you very much, guys. It was really informative, very helpful.
Thanks so much, Ian, and to the entire department. Somebody asked, we're going to put this up on the website, and feel free to contact any of us with questions. Thanks so much.